a lot of folks wanted to be here who could not, and including someone from the media said we'll be doing this. So I said that I would record it for their benefit, for transparency. So anyway, welcome Professor Ali Boehm and Sarah Lowry. This is the Kailua Neighborhood Board Kailua Water Quality May 2024 meeting. And I will call this meeting to order. It's 6:04. And as people may continue to drizzle in, I will be adding them. I have to minimize my screen for that. But I have announcements. I'm not going to read them because I just want to maximize our time together. Folks, I did put in the chat a couple of links of interest, including if you want to sign up for the brown water advisories from the Department of Health, an online version of the Healthy Yard Care book, which helps with stormwater runoff and best practices that we can do as individuals. And then there's another link up there. Some folks have been asking about stream maintenance, and basically like when the LA Pulu stream, what we know as the berm, when it gets opened to the ocean. So there's a schedule, there's a link online for that. So anyway, I would like to welcome Sarah. I had the opportunity to see her in person, and Ali. They were both here sampling. Sarah flew back yesterday, and on Tuesday we had an in-person meeting where she shared some of the preliminary results from the first 6 months of this study. And Sarah, what you may want to do is, for the benefit of folks who were not here in January, you may want to like introduce it in a broad way, because more and more people are coming to learn about this. So just for their benefit, introduce it, and then I'll let you take it away.
Great.
So welcome, and thank you both for being here. I know it's later in California, so thanks for staying up for us.
Yeah, thank you so much for organizing this, Leilani. So I will go ahead and share my screen. I have about maybe a 20 or 25-minute presentation, so there should be plenty of time for questions. Can you guys see my slides okay? Okay, I'll go ahead and get started then. And I can't see the chat or anything, so you might have to unmute if there's any issues. But today I'm going to be giving an update and sharing some preliminary results on the Castle Beach water quality study that we have been working on since January. And when I say we, I mean Dr. Ali Boehm, who is on the call, and I know a lot of you have met before. And Ali is a professor here at Stanford University in civil and environmental engineering, and she grew up in Kailua, so she has a strong connection to this area. She went to Iolani for high school, and she's been at Stanford now for over 20 years, and she's done a lot of water quality work over the years in various places around Oahu, Kauai, and Hawaii. And my name is Sarah. I am in my 3rd year of my PhD in environmental engineering. I have my bachelor's and master's in environmental engineering. And my research is on the human health implications of pathogens and indicators of pathogens in different water systems like stormwater, wastewater, recreational water. And then I'd also like to introduce Ella, who is a sophomore at Punahou, who has been helping us out on this study. And Ella lives near Castle Beach, and she's been really interested in learning more about the water quality over the last few years. And so she's done a great job sampling and taking measurements over the past few months for us. And so, like Leilani said, I'll start with, for anyone who wasn't here in January, or if you've just forgotten because January was a long time ago, I'll just recap sort of the goal of this study and the background behind it. And so we're interested in studying pathogen pollution specifically, so looking at microbiological parameters in the Castle Beach, Kawainui Marsh Canal area. And so what we've been doing over the past couple months is measuring the concentration of fecal indicator bacteria. And so fecal indicator bacteria, as the name suggests, are bacteria that indicate the presence of feces. They're not actually harmful themselves, so they're not direct pathogens, but they are indicators that feces and their associated pathogens are in the water. And so in Kailua, the Hawaii Department of Health also measures enterococci at 3 locations. along Kailua Bay. And they use those concentrations to make regulatory decisions, posting those advisory signs at the beach if you've seen those before. And enterococci are really commonly used across the US and the world for water quality. And so that's what we've used in this study as well. And so I'm sure, as most of you are very familiar with, there's a lot of potential sources or issues that may be contributing to water quality issues around this area. And so I've pointed out a couple of important landmarks on this map here, and I'll just briefly go through them. The blue star it's not quite accurate, but it represents the wastewater treatment plant discharge pipe that's off the Makapu Peninsula. And then that is fed by the two yellow stars, which are the Marine Corps wastewater treatment plant and the Kailua Regional Wastewater Treatment Plant. So they both discharge out there at that blue star. There also are a lot of cesspools, and those are the little red dots that you can see that are around the Kaimolino area in the north part of the bay. And then there's also Kawainui Marsh, which is I've indicated here by the green star, which has a lot of birds and things like that. And then the Hawaii Department of Health does regular monitoring of the beach at the 3 triangle locations, and they sample about every other week at 2 locations and weekly at the other. But importantly, they don't sample during brown water advisories. So there's not a lot of data, especially during winter. And so I'll give some specifics on each of these sources and why we're potentially interested in them. So the Marine Corps wastewater treatment plant has a record of exceeding enterococci counts. It was hard for me to find specifics, but I did find they were fined $250,000 for many exceedances from 2020 to 2022. And then the regional plant, we're now, I guess, approaching the year anniversary of all of those exceedances last April, when they had a lot of issues and a lot of beach closures resulting from that in 2023, despite a lot of recent.
Here we go, wastewater.
And then these 2 wastewater treatment plants, like I mentioned, discharge at the Blue Star off at the Makapu Peninsula. And then there's also the cesspools, like I showed you on the map. And for anyone who's unfamiliar with cesspools are just pits in the ground. They don't treat sewage, so they're different than septic tanks. And they pose a significant environmental risk, especially in Hawaii. Most are located near groundwater or the ocean. And so they could be a really significant source of nutrients, which can cause lots of environmental issues or of pathogens. And this little graphic right here shows that if the sewage isn't treated before it reaches the water, then it could infiltrate the recreational water and potentially get people sick. And there are about 90,000 cesspools currently in Hawaii. There's legislation that they all must be converted by 2050, but as we know, that's a really long time away. So these cesspools are still potentially problematic in the meantime. And then this is a map or a screenshot from the Hawaii cesspool prioritization tool. So just showing a different view of all of those cesspools where each one is a red dot. And so you can see this really high concentration near Castle Beach. And then the other potential source that we're interested in this study is the Kawainui Marsh and the canal. And so the marsh has a lot of water birds, and previous work in the United States has found that water birds can be really important contributors to fecal indicator bacteria concentrations. And so they could be a potential source of enterococci. But Ali did previous research back in 2011 with some of her students, and they found a whole host of pathogens that were detected in the stream, including Salmonella, Campylobacter, Staph, adenovirus, and norovirus. So we do have some evidence from quite some time ago that there were pathogens present in the canal as well. And we've also heard from residents and seen some of homeless encampments from unhoused people that are living in the marsh area. And so that could also be a potential contributor. And so the goal of this study is to try and answer the question, are fecal indicator bacteria found in the Castle Beach area from human fecal contamination? Enterococci are shed by warm-blooded animals, so they're not specific to humans. They can come from birds, animals like dogs, or humans. And in tropical areas, they can also grow in natural sand and grasses. And so when we're detecting enterococci, we don't really have a good idea of where they're actually coming from. But what we're really interested in is figuring out if they're actually from human feces, because they indicate the highest health risk as compared to these other sources. And so one way that we can try to figure out an answer to this question is to use this method called microbial source tracking. And so the idea behind source tracking is that different species will shed different genetic markers in their feces. And so you can test for a human genetic marker, and it's only found in human feces, and there's one for birds as well. And so the goal is to take these water samples and test them for these specific markers to try to figure out if there's evidence of human feces, bird feces, things like that, to get at answering this question. And so the formal objectives of this study are, first, to conduct a monitoring campaign to better characterize the fecal indicator bacteria levels at Castle Beach and the surrounding area. And then once we have conducted that monitoring campaign, we'll be trying to evaluate the origin of fecal contamination using this microbial source tracking methodology. And so I showed this back in January, but I wanted to remind everyone of the rough timeline of this study. So we first visited in January and did some initial sampling over a couple of days to try to establish some baseline concentrations. Then Ella has been working to sample twice a week at a couple of locations from January to late April. And then I had just left yesterday, and we were sampling for this microbial source tracking study for 2 weeks in April and May. And now that I'm back, I'll be working in the lab until hopefully December, maybe a few months later, depending on how everything goes, to work to process all of these samples. And so with our two objectives, we've now completed the monitoring campaign to measure fecal indicator bacteria, and the next step is going to be doing all of that lab work for microbial source tracking. And so today I'm going to share with you all of the monitoring data that we've collected over the past few months. And so I just wanted to characterize what exactly I'd be showing you. So I'll be sharing Enterococcus concentrations, which is the indicator bacteria, from the 3 sampling campaigns: our initial visit over a couple of days in January, our biweekly monitoring campaign that Ella conducted, and then finally this more intensive daily sampling from the past 2 weeks. And when I say that I'm reading Enterococcus concentrations, I just wanted to show you an example of what I'm actually doing, which is the picture here. So this is called an IDEX test, and you pour the water sample in with some reagents, and you leave it to grow overnight in an incubator. And then any of the wells or containers that contain Enterococcus will fluoresce over UV light. So this is a sample from a couple of days ago that had a good amount of Enterococci in it. And just as a reminder, Enterococcus is a fecal indicator bacteria, so it is not harmful, but it indicates the presence of feces. And as you're interpreting these concentrations, I've just put in here that the recreational limit that the DOH uses is 130 mpn per 100 mils. So you can think of that as a comparator when we're interpreting these values that I'm about to show you. So I'll start with the January sampling campaign. We sampled 8 sites, starting at the marsh and going all the way down the canal, ending at Castle Beach. And we measured enterococci, salinity, temperature, and turbidity. This is just a picture of the YSI probe that we use to measure temperature and salinity. And so here's just some summary results of our sampling campaign. The sampling sites are shown in this map here. You can see there's kind of a gap in the middle. We were just limited by access with private property, but we tried to get as many sites down the stream or the canal as we could. And we found that 56% of the samples we collected, we collected 32 samples in total, were above that limit of 130 mpn per 100 milliliters. And we had really variable concentrations ranging from below 10, which is the lowest we can detect, to over 1,000 mpn per 100 milliliters. And we didn't find any exceedances at the Castle Beach sampling site, which is where the DOH regularly samples. And so I wanted to show you all the actual concentrations for each sample that we took. So I'm going to introduce this graph first. This is 4 plots. Each of those plots is a different tidal event. So we have low tide 1, and then the following high tide, and then the following low tide, and the following high tide. On the x-axis, it's site. And so the sites from A to H, A is the marsh and H is the beach. So it's going down the canal. And then on the y-axis is the Enterococcus concentration. And then finally, this red dashed line is that limit of 130 that DOH uses. So I can show you what the results actually look like from this initial sampling campaign. Kind of a lot to digest, but you can see about half the samples are above that red threshold, and there's a lot of variability. So sites were pretty different depending on day and tide. There was a really high concentration. You'll see the 1200 bar on the 127 chart in the marsh. We're not really sure exactly why that happened, but the basic takeaway here was we found a lot, pretty high concentrations of Enterococcus, and there was a lot of variation, but it didn't seem to be statistically correlated with tide. So we needed more data points to be able to understand what was going on here. And so that's where Ella came in. And so Ella has been sampling from January to April for us, and she took 36 more samples. And so the two sampling sites that she sampled from are shown in this map in the small red points. One I'll call the Kailua Place site, and it's just north of all the cesspools around the Kaimolino area, near a lot of those on the canal. And then the other site is the Castle Beach site. And so she took 18 from each of those, and she measured the same things that we measured back in January. And so she found that at the Kailua Place site on the canal, half of the samples exceeded the 130 MPN per 100 milliliter concentration. And then I also just have some summary statistics. So the median concentration was around 83 MPN per 100 mils at this site. And then at the Castle Beach site, we only captured one event that was above the 130 per MPN per 100 mil threshold, but it was quite high. We found on one day a concentration of 460 MPN per 100 mil at Castle Beach. We also ran some statistical analyses, and we found that the Enterococci concentrations were positively associated with 24-hour rainfall. So when it rained in the previous 24 hours, we saw higher Enterococci concentrations, which I think makes a lot of sense. And a lot of us know that there's an association between rain, but it was good to see that in our data that we collected. There was also a significant association with salinity, which makes sense because we saw a lot more at the more freshwater site at Kailua as compared to the beach. And then I just wanted to show you the graphs. So these are all of the samples that Ella took at Kailua Place. So you can see, about half of them are above that dotted line, and there's a couple that are really, really massive. And those two big spikes followed two pretty large rainstorms that were in the area. And we can do the same for the Castle Beach site. And most of the samples were non-detect. So we didn't find any. They were below what our assay was able to detect for Enterococci. But you can see that one event, it was quite high in February. And so then we can move on to our sampling that I just finished conducting yesterday. And so we worked to sample around 7 a.m. and 7 p.m. every day for 2 weeks at 3 different sites. And we sampled at these times to try to avoid the sun. We know that the sun can kill Enterococci, and we wanted to make sure we were getting an accurate number of how many were actually there. And so we sampled at Kaha Park at the marsh, so the far end of the canal, Kailua Place at the same place that Ella was sampling for us, and then also at Castle Beach to kind of get a spatial gradient of all 3 of those sites. And so we grabbed, for this, we grabbed 1 liter water samples, so much larger water samples than what we were grabbing earlier. And we filtered them through a filter, and all of the filters now are back at Stanford with me, and we'll be using those filters in the lab for microbial source tracking analyses. And we also tested each of these samples for Enterococci using the same methods. And so I'll show you what those concentrations look like for each of the sites. This is the same type of graph you've been seeing, but now on the x-axis, it's just each sample. The labels are kind of confusing, but there's 2 samples a day for every day from the 23rd of April to the 7th of May. So you can see at Kaha Park, which is in the marsh, the concentrations are quite high. They almost always exceeded or above that 130 MPN per 100 mil limit. But there was a decent amount of variation. Sometimes it got really high above 600. So we saw consistently high concentrations at this site. And then as we move down the canal at Kailua Place, we see lower concentrations, but we're still consistently detecting enterococci here. You'll see the one event that was over the threshold occurred right after a rainstorm happened, which makes sense again. But we did consistently still detect enterococci at this site, just lower than the Kaha site. And then for the Castle site, for this 2-week period, we didn't capture any exceedances. We only found one day where there was even a detective enterococci. But I just wanted to say this is a really short sampling period, so we know that we're probably not able to capture all of the conditions that are actually going on at Castle Beach, like how we were able to capture that one really high sampling with Ella at Castle. And so we didn't detect any in this sampling, but it doesn't mean that there aren't enterococci concentrations at this site. And then while we were here, we also conducted an intensive spatial sampling campaign. So we went down the canal and sampled 23 locations in one morning. And so all of those sites are shown on the map here on the right. And then we also sampled a couple locations that we thought would be really useful to have for context in interpreting these results. So we actually took a boat out to the outfall at Makapu Peninsula and we grabbed the sample from there. And then we sampled 3 locations north of Kawainui Marsh along the Monowili stream. And so now I'll share with you the results of this spatial sampling campaign. So on the x-axis, 0 is like the farthest up in the marsh that we went, and 25 is down to the beach. And you can see here, there's a general trend, like what we saw in our previous results, that the around the marsh is higher, and then as you move down the canal, the gradient gets lower. And there were a lot of concentrations that did exceed that 130 per 100 mil threshold set by DOH. And then I also wrote out all of the results from the special places that we selected. We didn't detect any enterococci around the outfall. We weren't able to dive down to it. We just grabbed a surface sample and we didn't see any sort of plume, which is good. So we didn't capture any exceedances there. And then for the Monowili stream, north or above the Kawainui Marsh, at our farthest upstream point, which was kind of near the Monowili Falls trailhead that's now closed, we found a concentration of 40. In our middle site, we found the same concentration. And then at the site closest to the marsh, but just across the road from it, we found a pretty high concentration of 183 MPN per 100 milliliters. We also observed that that site closest to the marsh was really turbid, and it was located near a construction site. So that was interesting to note. And so that's all of our enterococci concentrations that we've gotten for this study. So our next steps are going to be analyzing all of the filters that I brought back from all of the water samples that we collected. And the goal is to try to answer this question of, is all of this enterococcus that we're seeing in water samples from human or animal sources? And like I mentioned earlier, this lab work will take several months. Hopefully it will finish up in December or January. And I look forward to keeping you all updated on these results. And I'm happy to take any questions or comments that you all have. Thank you.
I'll just say a couple of things. Sarah, you did a really wonderful job with the presentation, and I know you worked really hard. So thank you so much for all of that. And I know Jenny Lum is on the phone, who is Ella's mom, and Ella did an amazing job. And then my sister Alyssa and my mom Patty are also on the phone, and they also helped do some sampling. So, and then me and Levani's friend from high school took us out on the boat. So the community really rallied to help us. Friends lent, stand-up paddleboards. So people let us access their private property. So it was really just a community effort here. And it was a lot. It's been a lot of fun working with everybody. I wanted to say just a couple of things. So first, I think Sarah's absolutely right that we didn't capture a lot of contamination at Castle Beach, but I think what we have seen with all the data that she showed is that Kawainui stream definitely is a source of contamination. And I think the question is just, when does that stream impact the shoreline? And it happens during probably brown water events, and that's when nobody samples, or at least DOH doesn't. So I think understanding what is causing the contamination in the stream will be super useful for all of you. And then I also wanted to say that when Sarah does the molecular work to look for the source tracking markers, that's sort of an area of expertise that I have in my group, but we're also collaborating with a really amazing researcher at the EPA in the Office of Research and Development. And he's like the world expert in microbial source tracking markers. And I think, you know, working with someone from EPA is actually really helpful for all of us, including you guys, because if EPA is involved in the study, like it lends a lot of credence as well to the results. And it's something that, you know, our colleagues at Department of Health and other agencies like won't be able to, you know, ignore. I mean, they already have a lot of trust in us, and we've reached out to them about the project and they know about it, and we have really good relationships with them. So I don't think they would ever question results we bring to them. But it will be really nice, I think, to have collaborated with colleagues at EPA on the project as well. So we're hoping that, you know, when this is over, that we have a lot of like really good answers for you. And I've definitely seen in my experience working in this area, like different environments like this, where we may detect no enterococci, but we can still detect like human markers. So I'm very curious to see what Sarah finds with her work she's going to be doing over the next months.
Thank you, Ali and Sarah. That was, you know, such a good overview and very comprehensive. And I appreciated seeing all of the graphics because I'm a very visual person. And I did see Ella hop on. So thank you, Ella, for committing time and helping out with all of the sampling, and Alyssa and Patty. I'm going to open it up, folks, for questions here in the room and online. It was a lot of information, but I think Ali just did a really good job summarizing it. Do you folks have any specific questions or clarifications that you would like this team to ask? Yes, go ahead.
Yeah, hi, Sarah. This is Steve De La Pena. I saw you met you on Tuesday. I had a question in terms of the correlation between your findings and a lot of us who reported infections. Is there any indication yet about where that's going?
That's a great question. So I think it's a little hard because there's not, as far as I'm aware, any sort of like official database for reporting infections that's in place in Kailua. And so I've been trying to collect as much anecdotal evidence as I can. And it would be really useful if anyone in the community who hasn't talked to me before about that could send me if they've gotten sick swimming, if they have any details, because that is something that I can include even anecdotally in my report when I'm talking about interpreting these results. But as far as statistics, I don't think we have the database or the information necessary at this point to make a statistical correlation. Not to say that's something that we couldn't have in the future, though. But at this point, we're unable to do that.
Thank you.
I think a lot of people are wondering this, and it's also, I'm glad that you affirmed that, you know, even anecdotal data counts for something. So this is something I'd like to do, just in hearing a lot of like my paddling sisters and friends come and everyone. I mean, it's not just like one specific vulnerable population. Like we've known, you know, very young paddlers in their 20s who've ended in the hospital. So, because they had a small cut and it got infected. So just collecting those stories, I think, adds muscle, and I think it's really important. And I can share with you Sarah's address, and I'll email it out to the group. Okay, other questions? Let's go to Morley first, and then Doug. Go ahead, Morley.
Wonderful job, ladies. Great presentation. Very, very informative. Thank you. The data would seem to suggest, and I know this is a very limited, brief study, and it doesn't really look at the whole bay, but the focus really is Castles Beach and upstream. It would seem to suggest then that rain events and the upstream contribution as opposed to possible effects of the outfall is where our problem is. But then going further, could we say that it doesn't really seem to be tied to density and all the additional toilets that are now, flushing into Kailua Bay that never used to? That'd be a safe assumption, because if it is safe to say that, then there would obviously really have not been much change in your data over the years, because as I see it, the big change is the sewer. That means we've sort of had this problem all along. And I sort of do remember rain events as a kid being very dramatic. And you could really see the whole bay turn brown. We never knew there was that much bacteria out there, but it seems to suggest that there's not really much of a problem at Castle Beach, at least. The rest of the bay remains to be seen, but still hard to account for the infections and the brown frothy material that you see on a gusty trade wind day. No rain in sight, hasn't been rain for a long time. We see that a lot, and you can smell it. You really could smell it. So just curious what your thoughts are on that.
Yeah, I think that using enterococci, like Ali was saying earlier, it's not a perfect indicator. It's kind of like one of the better ones that we can use. And so this was our attempt to just kind of get an idea of what's going on. But I think what will be really useful in answering your question is actually getting the microbial source tracking marker analysis back, because that's independent of these fecal indicator bacteria concentrations. And like Ali was saying, she's done work where there's been no concentrations of enterococci, and we're able to detect human microbial source tracking markers, indicating that there are human feces contamination issues. And so I think it's useful as a starting place to look at this enterococci data, but we can't really discount the fact that maybe there is still human fecal contamination that the enterococci data just isn't capturing, and that we'll maybe see later. So I would say I can't really answer your question fully now, but hopefully once we finish the lab work, we'll be able to have a better answer, if that answers your question at all.
Okay, good. And then one final thought. I think I read this during some of the events last year. Apparently the plant the two plants into the outfall are authorized, or maybe just the Aikahi plant is authorized 16 million gallons a day of secondary, and they frequently exceed that. Does that pretty much, well, it's hard for you to say, but does that pretty much correspond to just rain events, or can that happen just in blue sky?
That's a good question.
Would you know?
Yeah, I don't think I have the answer for that. I will say that I know that the plant is under a mandate by the EPA to upgrade their UV, their disinfection systems, and some of their treatment systems. And I believe they have to do that, Leivani, I think but pretty soon, I think by 2025. And so they are undergoing a lot of upgrades right now at the plant from the exceedances that were caused by last year. But I'm unsure of if it was related to rainfall events. Maybe someone else in the room would have a better answer.
Okay, thank you.
Sarah, unless someone asks to refer to a slide, would you mind if we can stop screen sharing, and then I'll be able to see folks if anyone has their hands up or questions. Thanks. Doug, and then if there's anyone online, I don't see your face necessarily. I know that there's a lot of people who signed on. If you could just raise your hand, or I can try to go to the chat for questions, but we'll go to Doug first. If you can hit
I have two.
Can you hear him? He's in the back.
Yes.
Okay, go ahead.
I had two thoughts. One pertaining to the reporting of infections, and the other pertaining to the correlation between what has been recently discovered through this study at Castle Beach and upstream into Kawainui Marsh versus what's happening in the bay near the outfall. So to tackle the first one, and I'm tossing this out to the group here. There might be ways that we can work with the various healthcare organizations that are servicing our community, and on a voluntary basis, completely anonymous, find out how many people are coming, what days they're reporting, and these physicians are seeing infections. The information is there. There are probably some people who get a severe infection but don't go to the doctor, but most everybody does. So maybe there's something along that avenue to discuss and explore, or some other solution, but it seems that it's really important that we start monitoring pure data basis without any judgment, pure databases, how many people are getting infected and when it's that. And then we can correlate that with events that we know are going on. On to the second topic. The fact that this particular study is showing very little evidence in Enterococcus at Castle Beach. Well, that may or may not have anything to do with what's actually going on in the bay itself. And it may be that the operators of the two sewage plants know that since that horrible thing that they did to us last April, that there are a lot of people looking at this, and perhaps they now, with pun intended, are cleaning up their act, or maybe not. Or there could be factors such as tidal flow, current flow, various weather patterns that are affecting the presence or lack of presence of Enterococcus at Castle Beach. So that is something else that we need to think about and keep that in the back of our mind as we look at the results coming out of this very thorough test that Sarah's doing.
Yeah, this just related to that. One of the questions that I was thinking is, would there be higher concentrations along Kawainui because the water might be more stagnant and still versus Kailua, Kaimalino side, where there is a lot of mixing with the ocean water? So does that, from my layperson's perspective, does that make sense?
Yeah, no, absolutely. Like you can think of it as like a dilution, right? If you have like the ocean coming in and coming out in the really tidally influenced part of the stream, that could absolutely affect it. So I think our salinity measurements that we've been taking can maybe help us shed some light. I haven't had a chance to really dive into that data, but you can see there's huge fluctuations in salinity at the Kailua place site, and at Kaha, it's pretty consistent because the salt wedge doesn't really reach all the way up there. So that's a great observation and thought, and something that we'll definitely be exploring in the data.
I think I think that like a really interesting study to do would be a study, like a physical oceanography study of the fate and transport of the plume from Kawainui stream, but also Kailipulu. Like, you know, those would be really interesting projects to undertake in the future. It's not something that we aim to do in this project, but it would require like deployment of like current meters and temperature sensors, and it totally could be done. But it's not part of the present study. So, but it's something that I think should happen in Kailua. And I know that at UH, there's really amazing physical oceanographers. We talked to some professors at UH already about it who are super interested in collaborating with us, like on a project like that. But I think the physics part of it isn't part of like what we're undertaking with this specific project.
Thank you. Other questions? Do folks online have questions? I'll just quickly look at the chat here. Or Kelly, if you want to flag folks if they have a question, because one of my committee members just joined. Sonia says, when will you get information about human contamination versus other sources of contamination? And thank you, this was very informative.
Ideally, we'll have that, I'll have the lab work done by December. I did want to flag that working with the EPA means that we have to go through their internal approval process. So that does add some time. But like Ali was saying, it gives us that kind of credence to our study. So it might be a couple of months past that, maybe into like late winter, early spring of next year before the data is able to be shared. But that's the idea of the rough timeline.
We'll like push them to make it fast because I think it's important. So I might have to fly out there and threaten someone. But just kidding. Kind of.
Oh, it's always nice to have you back.
Fly out to
though.
Cincinnati. Fly to Cincinnati to EPA
not
and
Kailua.
threaten
Not the
them.
regional, Region 9. Okay. Eileen, I saw you online, but I didn't see you. Do you have any questions for the researchers if you're there?
Yeah, hi, no, very good presentation. I'm just very eager to see what the results are because we're always interested in what is human versus the animals and everything else that it could be. But no, I don't have any questions other than I'm waiting eagerly for those results. So
Thank you. Eileen is a physician, so she's really she's one of the strong warriors that we have advocating for clean water. Okay, Bob, do you want to, you have a lot of commentary. Can you just ask your question here?
I can.
You had a question.
Sure.
Oh, I was
type so
Or is that,
fast.
I was
Okay.
just going to say, I had a chance to briefly read your comments,
Oh,
Bob.
good.
I think I can respond to some of them. So first, we didn't make bird counts, but we are going to be measuring a genetic bird marker. So we should be able to get quantitative numbers of how many birds are at the site. I have pictures of all of the sites from when I was sampling. And so I could maybe try to count the birds from those pictures, but the idea would be that the genetic work would give us a quantification. And then we haven't.
Would you agree there's a general trend of more birds up towards the marsh and fewer down by the beach?
Absolutely. Yeah.
Yeah.
That is
Okay.
Yeah. And then the gene work be able to differentiate? So not between bird species at this point. There's only a general avian marker, so we can't tell the difference between the different bird species. We're not currently considering all of these animals that you've listed. Right now, we're certainly doing birds and humans, and we're thinking about dogs because we saw so many dogs around the areas, like in the dog park in Kaha and all the way down. It's something that we can consider. I'm not sure if there are markers for some of these animals like mongoose, but right now the plan is absolutely humans and birds because those are maybe two of the most likely culprits, and then maybe thinking about adding dogs. And then
Okay,
yeah.
You might also think of, I'd be surprised if they don't have one for rats.
I can definitely look into that.
and pigs also, because there's pigs in the marsh. And also one of the questions that we were discussing before we all signed on is the area up in the industrial area. We don't know if we don't know how their sewage is processed. But that goes down through Kapa'a stream and then comes down to Kawainui. So that would be interesting to know as well. Race station too, yeah.
Yes.
Other
And
questions?
I'd just like to make one more point before I leave, okay? They were saying that the plant leaves what, 1.6 million gallons a day? Was that it?
I think it's 16 million gallons. Is that the
16? Okay.
That's what I read.
A 1-inch rainfall will fill up Ke'elipulu Pond, 135 acres, 3 inches. And it'll make Kawainui rise, which is a square mile, about 2 inches. And if you do the math on that real quick, that turns out to 45 million gallons. So we always have to be careful when we're starting experiments that we don't kind of preconceive our hypothesis too much. After I've studied this problem for a while, I personally am convinced that the problem is not so much sewage, although that is a concern, that's valid, but a much, much bigger concern is all the water coming out the storm drain. So if you have the opportunity, you might just take a few samples from the storm drains or look at some of the available data for enterococcus counts that are coming out of storm drains and they are typically in the 10,000 to 20,000 range.
Absolutely. Yeah, I'm actually just wrapping up a project in Southern California on stormwater concentrations of pathogens and indicators in stormwater. So I definitely understand that there's a lot of nasty stuff that can be found in there, but that's great to keep in mind for this study. Thank you for sharing.
Yeah, if you want some of the data on Enterococcus counts in stormwater, just send me an email. I'll be happy to forward it to you.
Thanks so much.
Thank you, Bob. Other folks? Yes, go ahead, Judy.
I'm really thrilled that you're doing this because I live along the Kawainui Canal, about halfway between the marsh and the beach area. So I've always been concerned in the 50 years I've been there that something horrible was going to happen eventually. I wondered if you've had any contact with the indigenous group that takes care of the heiau, right where Manawili Stream enters Kawainui Marsh. If not, I have a connection with them and I'm sure they'd be happy to be involved in anything you might need. And you're also welcome to use my backyard for any sort of studies that you need to do. Thanks for all your work.
Thank you. We have not been into contact with that group, so it would be amazing if you could send their contact information.
Sure.
I would
absolutely.
Thank you.
Okay, there is a question from someone. Do you see that, Sarah, titled Court Observer?
I do. Yeah, that's a great question.
Can you repeat the question?
Yeah, so they're asking if we tested the sand because a lot of kids play and dig in the sand. That's actually something that Allie and I discussed, but I didn't have the materials to test sand samples in this particular study. I know Allie's done a lot of work testing Enterococci and sand, I think including in Hawaii, right, Allie? I don't know if you can speak to that briefly, but we didn't.
I haven't, but Tao Yan, who's a professor at UH, has done a ton of sand analysis around the island, and I would have to look at his papers and talk to him. I think he has done some testing in Kailua, but I'm not sure about Castles. But, you know, if we come back, that's something that we can definitely do. In California, these bacteria can definitely be found in the sand.
Thank you for that. Other questions? I know, okay.
Go ahead.
I was gonna say, Allie, I'll send the link out, but could you just say a few words? I did go to your website, your professor website, and it had a link to the, it was like a database or a dashboard for like pathogens in wastewater. Can you say maybe a few sentences? I think maybe this group might be interested, and then I can send the link out. Maybe Sarah, you can put the link in the chat for those online. Go ahead.
So like at the beginning of the pandemic, I got like wrapped into this huge project on detecting pathogens in wastewater that enters wastewater treatment plants for disease surveillance in communities. And we have a dashboard called data.wastewaterscan.org. And we do include the wastewater treatment plant in Kailua in our program. And we measure 3 times a week a large array of pathogens of wastewater from the community entering the plant, including SARS-CoV-2, influenza A, influenza B, RSV, human metapneumovirus, parainfluenza, Candida auris, hepatitis A, norovirus, rotavirus, and mpox. So our research shows that the concentrations of these pathogens entering the wastewater treatment plant are indicative of diseases in the community. And most recently, we've been working on detecting avian influenza in wastewater. And we did, this has been like in the news. We had like an article in CNN and in Reuters, and it's been like on Yahoo News as well, because we did detect avian influenza in some wastewater in Texas and in North Carolina. And we did test the wastewater from Sand Island in collaboration with Department of Health, and it did not have avian influenza. It was all non-detect, which is what we expected to find because there's no avian influenza on Oahu. So this is like not directly related to this project, but if you go to data.wastewaterscan.org, you can look at Kailua wastewater treatment plant and see what kind of diseases are circulating in the infectious diseases are circulating in the community in Kailua. And I'm happy, like Levani, if you want me to do like a whole, like a 15-minute talk on that, I can, next time I can like show people how to use the dashboard and stuff. If that's something you guys ever want, I would be happy to do that.
Yes, I see so many people nodding their heads. So there's obviously a lot of concern. Yes, as a public health person, yes, I would be very interested, and we can have you come back. I'm sorry I sprung that on you, but
Oh,
I
no,
didn't
it's
want to miss the
okay.
opportunity.
Like, I have to talk about it all the time, so it's no problem. But I would love the opportunity to like share more about the wastewater scan project with you all if you're interested. But it's not related to like this project directly, but it's still focused on pathogens.
Yeah, we won't steal your thunder, Sarah. It's okay. All right. We're about 5 minutes. Any other final comments or questions? Donna Wong.
Yeah, I just wanted to say, I'm not surprised that the counts are so high for Kawainui, because for decades that all the sewage from Mauna Wili, Mauka, went straight into Kawainui. And it took a whole community effort to get the city, to convince the city that to pipe it down, put it in sewage into pipes underground and completely avoid Kawainui. So the mat is huge and the water is stagnant, so there's no flowing. So it would just make sense that all those years that untreated sewage was flowing into Kawainui, that it, with heavy rain, that it would flush out.
Mm.
And with the industrial park contributing to it, because I believe they're either on septic or cesspool, no municipal at all, and all of the wastewater goes into Kapaa Stream, which goes directly into Kawainui.
Right.
Also, not to add and end on a downer, but it did come up at the neighborhood board that they were potentially considering the quarry as a future landfill site, which, you know, is of concern to us. So that's, you know, more for us to think about.
There was one there.
Sarah, any other final thoughts or on your next steps? I know it's late. Any kind of final thoughts that you'd like to conclude with?
Yeah, I just wanted to thank you all for being so engaged. It's been super valuable to hear all of your perspectives and get all this information from you. And it's really helped us guide our study, and hopefully will guide the interpretation of results once we get them back to you. And I think you all have my email, but please feel free to email me anytime with any questions or information you'd like to share. I'll be available. Thank you. Thank you,
Sarah. You can put your email in the chat. My caps were on, so I'm not yelling, but I put my email in the chat. If anyone wants to join our Kailua water quality mailing list, I send out 1 to 2 emails a month with just updates on everything related, or not everything, but as much as I can related to water quality in our community. Sometimes it might be 1 or 2 more if there's an opportunity for us to testify on legislation related to water quality, and presentations. I send out reminders because it's just such a golden opportunity for us to educate and inform ourselves and be empowered to do something. So thank you very much to Professor Boehm and to Sarah. Thanks for doing this work. We really, we can't thank you enough, just because I think it's the first study of its kind, and it's in a community that we love dearly and we want to protect and preserve for the future generations. And so if there's any other follow-up questions, please email me, folks. I'll make sure to get it off to Sarah or Ali, and we will conclude and adjourn our meeting at 6:58. Thank you, everyone. Aloha.
Thank you. Bye.
Good night, Ali and Sarah.
Bye.
Good night. Bye.
Very good. Wow. A lot scary, yeah.